Welcome to Tobacco University. Here we're going to be looking at some hair basics and getting some of the details for what we can actually determine when looking at a piece of hair and what components hair is made of. So first off, hair and fiber analysis. So in this case, it cannot be used to match a suspect with a crime scene. It can be microscopically analyzed for similarities, and comparisons can be provided to improve as forensics techniques improve. Keep in mind, hair and fiber both fall into the category of being class evidence. Now, hair and fibers for identification. Well, hair offers some benefits to forensic scientists because the hair resists decomposition, so it's more likely to be found at a crime scene. Hair may also indicate drug use or poisoning, and we can see that in analyzing the hair, different regions of that hair, we could find potential for drug usage in a certain period of time and potentially link that, or at least conclude that as part of the story for the crime scene investigation report. The anatomy of hair. So you're welcome to pause the video at this point and look at things in detail. We're going to be focused on the medulla, the cortex, in the outer portion, the cuticle region. However, realize that hair is embedded in the skin. It's all the way down to the dermal layer where the blood vessels are found. But we're going to be looking specifically at that hair fiber and some of the areas within that hair fiber. So first off, we got to look at the general structure of the hair. So on the outermost portion, we have the cuticle, and this has overlapping scales, and we can notice that here in the green region. Then we have the cortex, which is a protein-rich structure around the medulla that contains the pigment. And in that center region there, we have the medulla, which is the central core. In some instances, that may be absent, so it's important for identification purposes. The reason why I have the pencil here is we can think of this hair structure as a pencil, where the medulla being the graphite, or that center region, the cortex being the wood represented by the pencil, the cuticle being the paint on the outside, just to get you to understand how these layers interact and where they're located. Now that hair structure, again, what we're focusing on is kind of these different layers, and we could see that while the hair looks very thick in this instance, it's actually made of many small fibers, which is part of the reason why it's so strong for its small diameter. And again, just so you're aware of where it's located within the body, within the integumentary system. You can see here where it's located with some glands, where the hair matrix is located, and the hair bulb down here at the bottom. We're looking mainly here at the hair shaft and portions of the root. So this hair follicle, look at the follicle as part of the skin, which grows a hair by packing old cells together. It's attached on the inside and top of the follicle are sebaceous glands, which are tiny sebum-producing glands in almost all skin except the palms, lips, and soles of the feet. So here we're looking at the sebum being produced, and that's kind of what gives the hair some of its protective oils as well. The root of the hair shaft is part of the hair, not anchored to the follicle, and much of this is exposed at the skin's surface, and the rest of the hair, which is anchored in the follicle, lies below the surface of the skin and is referred to as the hair root. So when we're investigating a potential hair that may have been forcibly removed, we might want to identify or properly identify an area of the hair shaft compared to the hair root region, and there's that sebaceous gland tied right in, providing the oils again for that hair. Looking at a cross-section of the hair. So again, think of that same pencil concept. This is showing the different layers of the hair shaft, that medulla being in the center. Then we have the cortex here that may contain pigment granules, and then the outermost region being the cuticle. That cuticle being the outermost region, remember that is the covering of the hair, and we can see that here. It's formed by flat overlapping and hardened scales, and the degree of those scales again help us identify whether it's human hair or potentially some animal hairs, as we'll see in another video. But that hair cuticle is that outermost covering. Here is an example of it. If you remember back from that first one I showed you, it was the green outer area. And it's important for distinguishing between hairs of different species, but are often not useful in distinguishing between different people. So it might be important to know, oh, we found some hair. Well, is it the neighborhood cat, or is it someone that might be a human? So again, the cuticle can be used for identification purposes. When comparing the scales, you want to look at how many there are per centimeter, how they overlap, what's their shape, how much they protrude from the surface, what's their thickness, and is there pigment present, are all questions you want to ask when analyzing the cuticle. The cuticle classification, so they all kind of look the same. Well, actually, if you look very carefully, particularly with a microscope, there are different categories that they will fall into. And this, again, can help us identify what potential species that hair might be from, and can be used to eliminate, or again, looking at class evidence here, eliminate certain hairs that might be found that aren't of human species. The cortex, so now we're kind of looking at that next layer down. It's the thickest hair layer located in the interior portion of the hair between the cuticle and the medulla. It contains the hair pigments, or melanin, and pigments are absent though in gray hair. So as someone ages and gets gray hair, they're simply losing their pigments in the cortex region. That cortex specifically, the distribution of cortex is considered the most important component when attempting to distinguish between different people. Again, this is still class evidence, not getting to individual evidence, but still important for different people. Microscopic examination can also reveal conditions in shape of the root and the tip, and the cortex varies in its thickness, its texture, and also its color, which is kind of important for that identification purposes. Here we see human hair, and we have on the head, there could be on the body, it could be different areas we could be looking at, and we do see a difference in the cortex in the two images provided here. The cortex to pigment granule distribution. So this is both looking at a tabby cat, and again looking towards the end, most is closer towards the root area, and we can definitely see that there are differences in the cortex between these two. Keep in mind there's also a slight difference in magnification, so you're also seeing more detail in the cuticle region of the hair in this image down here. Getting to the medulla, that center portion, interior-most area of the hair. This is the most noticeable hair feature in many species. The medulla is that center region. So like the cuticle, the medulla is characterized by importance in distinguishing between hairs of different species. But they are often not useful in distinguishing between different people again. The medulla may vary in its thickness, connectivity, the opacity. That refers to kind of the structure. Is it broken into pieces? Is it all continuous? How much light is able to pass through? And keep in mind, it could also be absent in certain species as well. So again, very important when we're looking at class evidence is the medulla region of the hair. Medulla patterns, so there's three main characteristics. We could have a continuous, an interrupted, or intermittent. More specific, we could have fragmented, or it could also just be absent, and that's basically relating into no pattern, but would be important to note. The classification of the medulla, so we could see here again, we just have different classifications for structure and pattern forming at the margins. And again, this can help us use this information as class characteristics of the hair samples found and collected at a potential crime scene. There's also something called a Medulla Index, and this describes the thickness of the medulla layer. It's calculated by measuring the diameter of the medulla and dividing by the diameter of the hair. So here the Medulla Index for human hair is generally less than 1/3, and the Medulla Index for animal hair is usually greater than 1/2. So this can help be another form of quantitative analysis when we're trying to classify different hair. Using the Medulla Index is one method. So as you can see, there's a lot of different types of ways to kind of look at hair, analyze hair, and understand hair. And hopefully this provided you with a little insight to all the options that are available.